So everyone, welcome to today's seminar. Thank you so much for joining us. My name is Cinnamon Moffett, and I'm the Research Program Manager at Hatfield Marine Science Center. And I just wanted to thank you so much for joining us. And we're going to get started here in just a second, but I wanted to just do a few little logistics. Wanted to let you know that your cameras, mics, and screen shares have been turned off at this point. And if you could help us out by keeping them that way, that would be really great. We would love for you to ask questions during this talk today. You should be able to find a chat box either at the bottom or the top of your Zoom screen. And today's speaker is willing to take questions at any time. So please, if you have any thoughts when he is speaking, please put your questions into the chat and he'll answer them when we're talking about whatever the relevant question is at the time. I also wanted to let everybody know that this is being recorded. And so if you are interested in watching any of the past seminars or this one in a few days, you can visit HMSC's website, go to Visit Hatfield, and you will see a past seminars page, and a link to today's seminar will be posted there. Also wanted to let you know, if you're taking this for credit, Michael Banks is listening and is online. So if you have any questions for him, you can put those in the chat as well. Quick little promo for next week. Next week, we have Renee Bellinger from the University of Hawaii in Hilo is going to be talking to us about animal migration and how animals know where to go. So excited to hear a little bit more from Renee about her work. And again, if you need to know any more about Hatfield's events, you can go to Hatfield's website, scroll to the bottom, and there's a calendar of events there. But with all that aside, what's important is what we've got going on today. Today's speaker was actually invited today by our Hatfield student group, the HSO. So I'm going to have Karen, one of our students, introduce today's speaker. So Karen, if you want to take it away.
Hi, everyone. As Cinnamon said, today's speaker, Dr. Eric Archer, is student invited. We are so thrilled and honored, and the stoke level is very high to have Dr. Eric Archer here speaking with us today. He is the head of the Marine Mammal Genetics Program in the Marine Mammal and Turtle Division of NOAA's Southwest Fisheries Science Center. Before that, he did his PhD work comparing morphometric and genetic variation in striped dolphins at Scripps, where he's now actually an adjunct professor as well. His recent research on topics include population structure and taxonomy of small dolphinids, large whales, developing algorithms of acoustic classifications of cetacean species using acoustics to better understand cetacean population structure, and a whole host of many other things that just are super fascinating. He's also sent a list of hobbies, which I just love because they're very interesting, and they include aviation, martial arts, playing the bass, hiking, and kayaking. So it sounds like Dr. Archer is very busy, both in and out of the lab, which we have a lot of respect for. And so with that, I want to turn it over to Dr. Archer to tell us all about making lemons out of lemonade, which is nice and hopeful for this year.
Thanks, Karen. Thanks so much, Karen and Cinnamon, for inviting me here today. I'm really excited to give this talk, to share this research. It's been one of the projects that I've worked on that I've actually I've had fun throughout the entire project. I think it's a really interesting one. I'm just happy to be able to share it to this group with this group. I wanted to acknowledge the co-authors on this paper, primarily Jill Braulick, and this project is really her baby. She's been working on this for many years, and I'm just a hired number mover on it. So I kind of feel honored to have been able to, that she entrusted me with her data. And so I'll just be presenting some of the work we've done putting this paper together. And and she's was been supported by many other researchers in the region, gathering samples, doing observations, and I just wanted to acknowledge the amount of effort that's gone into pulling this data together. And like I was saying, because it's hard to share somebody else's story. I'll try as hard as I can to answer questions about the systems, but just saying straight out that I'm not an expert on river dolphins, especially this group. But I'll try as I can. We also have I think Tom Jefferson might be on the line as co-author, and he's did a lot of work working on the systematics and nomenclature of the group. So if I can't answer a question, I'm sure Tom can back me up on it. So I'll start with a general overview of the system. Currently, there are 2 subspecies in the genus Platanista, the South Asian river dolphin. Platanista gangetica minor is found in the Indus, and then Platanista gangetica is found in the Ganges, Meghna, and Brahmaputra river systems. In this talk, I'll pretty much just be referring to Indus and Ganges as a shorthand for the 2 river systems. These river systems are thought to have been distinct for about 45 to 55 million years as a drainage basin for the Himalayas. It's thought that the Platanista first colonized the Ganges and then dispersed into the Indus during some capture events, river capture events in the last million years. Indus dolphins, as I've said, are endemic to Pakistan in the Indus, and Ganges dolphins are endemic to the Ganges and in India and in Bangladesh. And because of that, it's difficult to study them because the 2 countries are regularly on a war footing, and so it's hard to find people that can make observations of both types at the same time. The population sizes are relatively small. There's thought to be about 2,000 in the Indus and about 5,000 in the Ganges. There's also been a very drastic range reduction in both river systems, about an 80% decline in population in range in the Indus and about 30% in the Ganges. And those of you that know anything about the Ganges and India know that it's a heavily used river system, and its use is increasing. And so there's a lot of anthropogenic effects on dolphins there. So both of these forms are under large threats, and conservation is a real issue. The 2 species, or 2 forms, are in the genus Platanista, and that genus is a very old form. It diverged from the rest of the Odontocetes, not including the sperm whales, somewhere between about 30 million years ago. It was thought to be more widely spread, but currently exists only in these 2 river systems, and as sort of retreated to these 2 river systems with a relatively recent divergence of the 2 forms. They're closest, they are sister to the rest of the Odontocetes, just outside of the sperm whales. So there is no other close relative to Platanista within the rest of the cetaceans. Their taxonomic history they have a long and varied taxonomic history where they've bounced between species status and subspecies status. They were first defined or described in the early 1800s. The original description was actually of Delphinus gangetica, but later was assigned to Platanista gangetica. And in 1853, there was a mention of another variety from the Indus, which was termed minor. Later in the century, or in the decade, that form got the name of Platanista indi, and that was the original description for the form from the Indus. So at this point, there were now two species, gangetica and indi. However, later on, Anderson, looking at skull and skeletal morphology, determined that there wasn't enough difference for species and demoted them to subspecies gangetica and indi within gangetica. Time passes, and in the mid-70s, a lot of work by Pillory and Gear resurrected the two species as gangetica and indi as species status based on differences in morphometrics, blood proteins, both external and internal morphometrics, blood proteins, and lipids. In the mid-70s, Van Bray rediscovered the minor variety, and which had precedence over Indus, and so that species was then put into Platanista minor. A little bit later, after a few events, there was a reanalysis of the species status, primarily Reeves and Brownell, looking at what had been used to determine them as to designate them as species, and realized that the sample size was small, and it was more descriptive, and it was not composed of enough adults, so they were demoted to subspecies, which is where Dale Rice left them in his handbook, and that's where we are today. As having two subspecies, gangetica and minor. So the goal of this project was to take all of the available data on these two forms and ask a question: are they subspecies or are they species? And so it's this mix of data, and we're trying to take these different lines of evidence and ask something about discrete structure along, what we recognize as the continuum. Jill published a paper in 2015 looking at some control region sequences, but that wasn't definitive at the time, although it was very compelling. So we did some reanalysis of that data. She also spearheaded a survey of external coloration and collected data on external growth, or body length and weight measurements. So we did some growth modeling. And then the big data set was a set of skull measurements. Some of them had been aged, so we could actually look at skull growth. So for the rest of this talk, I'll be kind of going through the evidence from each one of these lines of evidence for, what they tell us about either subspecies or species status. So let's start with the genetics. This is all more a recap of Jill's paper. So this map is from Jill's paper, just showing the sample sizes from the two river basins, fairly widely sampled from the Ganges-Brahmaputra basin, and a fair number of samples from the Indus as well. However, these are not fresh tissue samples. They were sampled from skulls, which were deposited in museums around the world. So she was only able to sequence some mitochondrial DNA. And this table shows the variable sites for two sets of sequences. One set was a 458 base pair sequence, and the other was an 858 base pair sequence. And there is some overlap here, but the numbers don't add up because not all samples could be sequenced for the 858. The general picture was that there were 6 haplotypes if you looked at the 858, 3 in the Indus and 3 in the Ganges. Those six haplotypes were separated by 5 variable sites, one an indel, one a transversion, and one a transition, and they're sort of listed here in gray. If you just looked at the 458 base pairs, there were 2 fixed differences, 2 transitions that separated the 2 river basins. So relatively strong differentiation, and 5 diagnostic sites across this set of sequences. They created several phylogenetic trees, and the issue that they ran across was that because of the distribution of variable sites, there was very little phylogenetic resolution. So you do not get clear reciprocal monophyly of Ganges dolphins and Indus dolphins. You get these polytomies. And that's because the tree is operating on the haplotypes, and it really doesn't pay attention to their original location. So it's just trying to reconstruct the pattern of descent of the haplotypes, and the nature of these variable sites means that it's actually quite difficult to do that, even though you have a large number of fixed differences. However, the Bayesian tree was able to estimate that they diverged, or they all had a common ancestor about 550,000 years ago. So we at Southwest and some other colleagues that we've been working with in 2017 had been looking at how do you determine if you have species, subspecies, or populations if you're only working with genetic data. And we put this work together as a multi-year project into a special issue in Mammal Science. And one of the cornerstones of this work is in one of the papers by Barb Taylor, where we developed a set of guidelines based on empirical or currently recognized populations, subspecies, and species. And in this figure, the red dots or red triangles are currently recognized cetacean populations. The green squares are currently recognized cetacean subspecies, and the blue circles are pairs of recognized species. And the axes here are 2 metrics that we found to be useful for determining the taxonomic status. The x-axis here is Ney's net divergence, and it's a measure of differentiation amongst sequences that gives you a sense of how far down and sort of evolutionary trajectory things are. This is on a log scale. So the more time that passes and the more net differences that accrue, the further to the right 2 pairs will be. The y-axis here is percent diagnosability. And this is a measure of how well you can assign an unknown specimen to its taxa of origin. So this is kind of what we would refer to as the museum problem. You have a specimen at hand and you've got some training samples from museum that are vouchers, and you create a taxonomic key. How well can you take that unknown specimen and put it into one pile or the other? You expect to have some set of characters that will allow you to correctly diagnose any individual from the species pair 100% of the time. That is no error. For subspecies, you expect there to be some error, but not too much. And for populations, although they have frequency differences, you don't expect them to be highly diagnosable. You expect there to be considerably more error. And so that's what we see when we look at these empirical examples. And so now the question is, where does Platanista fall on this? Well, here's a summary table for both the 458 base pair and 858 base pair sequences. And if we look at those two values, we see that Platanista kind of falls about with the other subspecies. Its net divergence DA is just over the threshold that was set for subspecies, but not over the threshold that was set for species. But it is fully diagnosable. That is, we can tell individuals apart. So just based on genetics, you would say that this falls into the subspecies category. However, we recognized in that special issue that you could have other lines of evidence. So if you had high diagnosability and you had something else that indicated that there was no there was reproductive isolation or other differentiation, then that would push you over this line into the species category. So let's look at the other lines of evidence that we've had collected. So one of the other group data sets was external growth. So this was a data set of body and length and weight measurements for about 126 individuals. And what it did was to fit a simple Bayesian growth model to these. And we found something very interesting in this. This top row, we have the two river basins, and they are colored by sex. On the bottom row, we have the two sexes, females on the left, males on the right, and they're colored by river basin. It's the exact same data. The thing that we found that was relatively interesting was that in the Ganges, there is sexual dimorphism in the growth. That is, females tend to grow to a larger asymptotic length for a given weight than males do. And that was not true in the Indus. Indus dolphins, females and males that were about the same weight were about the same length. And in general, the Ganges females were grew to a larger size than either the males or the females in the Indus. So if you have a Ganges female that is sort of fully grown, she's going to be much larger than the largest of all possible kind of Platanista individuals. So that was kind of surprising. Essentially, between the two river basins, there was a difference in sexual dimorphism. Jill also did a survey of color pattern, and although it was very subjective, it still had some information in it. Sent it out to 42 people, got 33 responses back. 26 of those were very familiar with Ganges dolphins and 11 very familiar with Indus. Unfortunately, as I said before, few are able to really make good observations of both. So she didn't have a lot of crossover, but the big take home was that about 88% of the ones that are familiar with the Ganges, they were described as being either gray or grayish brown. while there was more color variation in the Indus, most of those were described as being brownish gray or grayish brown. None of the Indus were described as being gray. Now, there's, of course, a lot of question about differences in turbidity, light coloration, and it's hard to compare these, but it is an indicator that there might be some color, just some general color difference between the species. So moving on through our data sets, we also, the biggest data set we had was the skull data set, and is a similar sort of sampling scheme and sampling distribution amongst the river basins. One thing, if you've never seen a Platanista skull before, they're pretty amazing works of machinery. They, like other river dolphins, they have this very long rostrum, but the thing that they're noted for are these maxillary crests, these large bony plates that kind of grow out the edge of the maxilla and cover the nasals and the proximal end of the rostrum. They're most likely related to sound production. They're super cool. The issue with them when it comes to morphometrics, of course, is that because they fold over and cover these areas, it makes getting some traditional landmarks and some traditional measurements really difficult. But Jill and her colleagues took as many as they could and tried to capture various measurements on those maxillary crests. There were a total of 18 measurements taken. And then we took several of these measurements and made ratios out of them. For instance, we made a ratio out of the height of the maxillary crest to the width. We made ratios between the left and the right maxillary crests. And we also did ratio measurements where we scaled the long measurements of the skull and the width measurements of the skull to either this standard condylar basal length, the length of the skull, or the width measurements being scaled to the preorbital width. And in later slides, you'll see those as being measurements that are either .cbl or .prow. So in total, we had some 46 measurements that we could play with. We also had upper and lower tooth counts, and the distribution, and then several of the skulls we actually had ages from the teeth for. So although relative to other morphometric data sets, it's a pretty small sample size. For this species, where it's really hard to get samples, it was a pretty impressive collection. So the first thing we did was to do a similar set of Bayesian models, growth models for the skull measurements, where we modeled each skull measurement relative to age this time instead of weight. And we see the same difference in dimorphism between Ganges and Indus. In all of the length measurements, females were longer for a given age. So it's not just a difference in the weight, just in the difference in their weight relative to length. For instance, if we didn't have this data, one possibility would be that Indus dolphins are heavier for a given length. It's that as they grow, and because this axis is age, female Ganges dolphins are actually longer, and we don't see that in Indus. There is a caveat here where we only had one really old adult Indus female, and as you can see, sample sizes are quite small. But the fit of the curve, it was pretty good, and so in the Bayesian analysis, the uncertainty around the posterior was actually relatively small. You would want more samples to really verify this, but since it fits with the external measurements, I think this pattern sort of holds. We then do sort of standard cranial morphometrics, and there's several things that jump out when we look at kind of each measurement by itself. The first thing is that there really was no diagnostic measurement. There's no single measurement or ratio measurement that showed kind of fixed differences between the two river basins. There's nothing that we said, if it's greater than this, it's definitely Indus. If it's less than that, it's definitely Ganges. There was always some overlap between the measurements. We see the sexual dimorphism in the length measurements, but it's only in the rostrum measurements. So this is condyle basal length, and these are those very long Ganges females. When we look at other measurements, length measurements as a proportion of the length of the skull, we don't see that difference. So this is length of the lower mandible, and the females do not show a sexual dimorphism in these measurements, and that means that it is the rostrum, the upper rostrum, that grows in Ganges females, not kind of just the entire skull is longer. It's just the rostrum that seems to get longer with age in Ganges females. Most of the differentiation that we see, although there's overlap, is actually in these ratio measurements. This is the ratio between the left and the right height of the maxillary crest. This is the ratio of the area of the maxillary crest. This is the width of the maxillary crest as a proportion of essentially the width of the skull. And then this is the ratio between the length of the orbit from left to right. And you can see that the differences are relatively small. The biggest difference, though, is between for things that involve the maxillary crest. And we think that that might be informative. So we then come to looking at this in a multivariate sense. And there's typically two kinds of multivariate analyses that we like to do. One of those is feature extraction or ordination. This is your standard principal components analysis. The other one is looking at diagnosability, again, using, and here I like to use random forest. This is trying to see how well we can classify skulls. The problem with this is that both of these methods require complete data sets. And this skull data set was anything but complete. These skulls, as you can see, they're very fragile. They tend to break in preservation or transportation over time. So it's really hard to get all the measurements that you need from these skulls. So this figure kind of shows the distribution of missing data in the skull measurements, just the base skull measurements across all of the samples in the data set. The little gray areas are missing, and then they're colored by whether green Ganges and orange for Indus. So in order to do these analyses, we had to come up with a method of either removing certain samples or removing certain measurements. And we wanted it to be systematic. So this kind of gave me an opportunity to kind of come up with an actual algorithm to do this. And we called it the Iterative Missing Data Optimization Algorithm because it was about 2 in the morning when I finished this, and I couldn't come up with anything catchier. And the idea is to look at patterns of missingness in the data and to try to create a set of data sets based on patterns of missingness. And we start with all of the skulls that have no missing data. So that is this group, and we're going to call that DI. And so we're going to go through many iterations, and this is going to be the data set for the first iteration. So we're here, I is 1, the first iteration. We then group all of the other skulls that are not in that data set based on their pattern of missingness. So here you can see that there's 2 skulls that are missing for this measurement. Here's another 2 skulls that are missing for this measurement. Here's 4 that are missing for this one. So if, in fact, these 2 sets of skulls have the same pattern, so this would be one group. So you do that for all of the patterns of missingness. And then you want to choose one of those groups, but then you have to pick some criteria. And for this analysis, we essentially want to maximize the power of the analysis. So we first want to make sure that the group that we're adding has, as best as possible, both river systems in it. It should also have the river system with the smallest sample size in the current data set. That is, we want to maximize the balance of sample sizes across the 2 river systems. Also, I want to look at the ratio of the skulls that I'm adding, number of skulls I'm adding, to how many measurements I'm excluding if I choose those skulls. That is, I don't want to choose only 3 skulls if it means I have to exclude 8 measurements. And then finally, I want to choose a group where if I exclude a measurement, I'm getting rid of measurements that are not correlated, or that are correlated with measurements I already have. So I can throw out measurements that aren't adding much information. So I kind of walk through these 4 criteria, and we'll choose a set of skulls in a group that maximize these criteria. Those get added to the data set, and that becomes my data set for that iteration. And then I exclude those measurements that are missing in those skulls, and we repeat the process. So you end up with a set of data sets. And then the question is, what's the best data set to use in the multivariate analysis? And we realized that we could order these data sets on a figure. So here the x-axis is the number of measurements in the data set, and the y-axis is the number of skulls. And in the bottom right, we have our first data set, and that walks its way through those iterations where we have all of the skulls that are not missing any data, and that walks its way up to all of the skulls and how many measurements are not missing for all of the skulls. And what we choose as the optimal is the data set that is essentially the farthest away from a straight line between the 2. So this is the data set that we would use for in the upcoming analyses. And we have multiple data sets. We had data sets where we had all of the skulls in things that we had identified as adults. And then we have different measurements. We either have those standard absolute measurements, ratio measurements, or a combination of those measurements. So here's the result of the principal components analysis. And this is a standard principal components analysis. It's the first 2 components. And it's generally thought that if you've got a large amount of differentiation, it's going to pop up on these, and it's really not true. There is no structure on these first 2 components. This is these 4 data sets. This is the absolute measurements using all skulls. This is absolute measurements using just adult, ratio with all skulls, and ratio with adult. And the first axis is 94% for the absolute all, and that makes sense. This is an axis just showing the size of the skull. And then you start to quickly drop down to 2% on this. And this is just, we have skulls of different sizes. It looks like that there's maybe a few more indus on the right side. but if you actually run statistics on this, it's not significant. And the same holds true kind of going across. There's not a lot of structure. When we look at the adult skulls, those really large females pop out here. And we'll see that in the next slide, why that is. But otherwise, there's just no structure. So we actually have to look component by component on these. And that's what we've done here. These are all of these significant differences for individual components for the absolute measurements. Ganges is in green on the left, Indus is on the right. And this is these bar charts show the relative loadings on that component. So for PC2 here, this is really a measure of the length of the left orbit. And we see that Indus being slightly more negative tends to have a slightly, this actually is reversed because this is negative and this loading's negative. That's saying it actually has a larger orbit. But what we actually see throughout most of these is that there is a difference in the maxillary crest measurements. So here we have width of the right and left maxillary crest, and here's the height of the left and right maxillary crest. So this component is actually a contrast between width and height, and we can see that Indus tends to have higher maxillary crest, taller maxillary crest, while Ganges tends to be wider. And similarly here, we've got length at the base of the maxillary crest opposing height, and we can see if there's a significant differentiation between Indus and Ganges on that. If we look at the ratio measurements, the principal components of that, we see a similar thing. We see a lot of contrast between maxillary crest measurements. And the general thing that we get out of this is that Ganges seem to have more of an elongated oval maxillary crest, where Indus has more of a round maxillary crest. These are significant differences, but again, they're not diagnostic. So then we go to diagnosability. This is how well can you classify, how well can you actually tell apart different skulls. And ran a straight random forest on using all of the characters on these optimal data sets. And what we end up with is about 80% diagnosability on the skulls. Not super high, but not super low. We have, for adults, a much smaller sample size, but even all skulls, it's about the same. So it really doesn't matter if we use adult skulls or if we use all skulls. We don't have high diagnosability from just the skull measurements. Another thing that we get from the skulls are the tooth counts, and often these can be diagnostic for between different species. So we fit just a straight logistic model, and here we have a greater upper tooth row count and greater lower tooth row count. And between the two species, it's about a 1 tooth difference. This is the difference between the two. And if you use the estimated kind of cutoff of 31 and a half teeth, you can actually get about 92% correct classification of the skulls. That is, you can correctly classify Ganges or Indus skulls about 92% of the time. Indus skulls tend to have 1 more tooth than Ganges, 1 to 2 more teeth than Ganges skulls do. So it's actually more powerful than the skull measurements. And if you combine both the tooth counts and the skull measurements together, you still get about 93% correct classification. But in all of this, and you're going to be mad at me at the end of this, because I've totally buried the lead. We've gone through all of this statistical rigor, trying to make sure that we've got the best data set. And the actual answer is one that even a 5-year-old, as Jill can attest to, can tell the difference. It has to do with a feature in this area of the skull. These are the frontal bones here that are coming up and sort of meeting, and there's a suture here at the top of the skull. And that frontal suture has a consistent and diagnostic feature between Ganges and Indus dolphins. In Ganges dolphins, that suture tends to be protruding and goes to the left. In Indus dolphins, that suture is flat and not protruding at all in some cases. And this is in absolutely every skull that's been examined, even shows up in fetal skulls. So it is 100% diagnostic. So the bottom line is, if we were just looking for a diagnostic cranial feature, I didn't have to do any math. I could have just called a 5-year-old over, had him touch a skull, and you can tell the difference. It's pretty striking. So let's summarize what we have as differences between these two forms. To genetics, we've got 5 fixed differences in the mitochondrial DNA, and we see we have an estimate of a time since the most recent common ancestor, 550,000 years. There seem to be some coloration differences, but there's still some question in that. It's not very objective data. It's still it's one of these things that you would want to get more information from. There are differences in sexual dimorphism, both externally and internally, where there is sexual dimorphism in the Ganges, and there doesn't seem to be any in the Indus. And then finally, that sexual dimorphism seems to be related to the length of the rostrum, but then there's that frontal suture that is a morphometric feature that you can easily tell the difference. There are almost diagnostic tooth counts, and definitely significant differences in the overlap, and then potentially in the shape of the maxillary crest. It's the kind of thing that I think, with the way this traditional landmark morphometrics, it's hard to capture. It's the kind of thing you'd want to get more sort of higher power, say digital morphometrics, 3D or 2D geometric morphometrics, to really quantify those differences and visualize the differences. But I think there's strong evidence that the maxillary crests are different shapes. So if we think about this in terms of species concepts, we can reorganize this and look at this typical biological species concept that is focused on reproductive isolation and evidence of reproductive isolation. Clearly, we can't do the experiment of taking an Indus and a Ganges dolphin and trying to make fertile and viable offspring, but there is circumstantial evidence. They have been physically isolated for a long time. There's differentiation that's arisen in multiple characters that it's hard to believe that it would arise if there was significant gene flow. And then differences in sexual dimorphism, which if you think that could be related to reproduction, could mean that there's like prezygotic selection. If you look at it from the phylogenetic species concept, we have 6 diagnostic characters, the 5 fixed differences in the mitochondrial DNA, and then the 1 frontal suture. And the interesting part about that, though, is that we do not have reciprocal monophyly, which is often used as a characteristic of the PSC. But that's more because we don't have the resolution in the tree because these are such old lineages, and it makes it difficult to polarize these characters. But the other side of the phylogenetic species concept, that diagnosability, is there, and it's there very strongly. So taken together, we sort of can separate this out, and I think it strongly argues that these two forms are species. And we've submitted a paper to Marine Mammal Science. It's back in reviews, and hopefully will come out next year. And then we'll see if it's accepted, and then that'll change the taxonomy of this group. This work has been based on a lot of different data sources, but relative to some other analyses, it seems in some cases the sample sizes are small. I just wanted to point out that this is a really difficult group to sample. So the kind of reinforcing of these different data sources, I think, makes a strong case, and it's unlikely that there's going to be kind of further samples to increase the sample sizes. There is a project that is, Jill was just telling me, they're starting to form a project to look to sequence the genomes. There's already a genome, I think, of the Indus, and they're going to sequence the Ganges, or it's the other way around. And that might give us a bit more information. But in terms of getting actual samples, I think actual physical samples, that's unlikely to happen. So we have to make the most of what we have, and I was really happy to work with this because I think that's what we tried to do with this. So just want to acknowledge several of Jill's colleagues and coworkers and people that have helped us on this project, and the funding sources that Jill had to do this work. And I'm happy to take questions.
Thank you so much, Eric. For everyone online, go ahead and put any questions you might have into the chat box. And while you're giving just a second, I'm just curious real quick, Eric, if your data poor analysis techniques have been tried on other species yet?
No, I just made them up out of thin air, and they haven't been published yet, so we'll see. This kind of iterative process, it was very kind of empirical. I think it will, I think it's the kind of thing you want to do when you've got data sets like this that have a lot of missing data, and you have to think about what the question is. What is that final data set you want? So I can see it having utility in with other questions, and hopefully it gets refined and made better once this paper comes out.
Nice. And it seems like we're getting a couple of questions in the chat box, which I think you can see, correct?
I can. So from Michael, is there any information on the viability of hybrid mating between Indus and Ganges, or viability of offspring? As far as I know, there are, I can't imagine that there would be two identified hybrids. They are, physically separated, geographically separated, and they would have to travel around India outside of fresh water in order to actually physically mate. And so I don't know, I doubt that there have been anything I don't think that's been observed. And then from Jeff, how do the two river dolphins differ behaviorally? I can't directly answer that, as I've not observed either one of them. I do know that they are very difficult to to see and to observe. It's in murky water, and it's one of the reasons that Jill had to resort to getting genetic samples from museums, because they're difficult to sample, to biopsy, because they don't surface in predictable ways. But I don't know that anybody actually knows how they differ behaviorally, because you'd have to be able to observe both of them and have somebody that could do both of that, and that doesn't seem to be an easy thing to do, just given their distribution in countries that aren't that friendly.
So I had another question, Eric, that might be another one that you feel uncomfortable answering, but can you go back up a little higher and just talk to us about the policy impacts between having these two be two different species versus being subspecies?
I can talk about it in general terms. Generally, if you have, essentially they would now be species endemic to a country. So you would have the Indus dolphin being endemic to Pakistan, and the Ganges dolphin is endemic to India / Bangladesh, and that aside from a sense of national pride, which that typically they seem to have for their forms, having a species endemic to your country then enhances the conservation goals, because then especially with their habitat loss, their loss represents a bigger biodiversity loss. So you have more effort at conservation. It's not that the effort should be small if they're just subspecies, but it just sort of ratchets it up to another gear when you've got an endemic species that's being lost. And if it's down at the 2000 level, it becomes kind of the next one's down after the vaquita. It's a really a species at risk.
Thank you. I think you had another question from John.
Yeah, so the obvious ones, are there other cetaceans and marine mammals with that geographical and temporal isolation that are similarly low diversity genetically? So the obvious one is the vaquita. It's down in the 20s, 10s in the upper Gulf, and there's just a paper that Phil Moore in our lab published on its genome that shows that it is very low diversity across the genome, but it's been that way for a while, for many millions of years. And so the point of that paper was to show that if you remove the causes of mortality, the fishery and the nets, it should be able to recover, because there's not as much worry about inbreeding depression. And so low diversity, there's still the jury's still out on what is low diversity and how we actually measure inbreeding, and how much low diversity is really a risk in things that have been that way for a while and that are restricted.
Nice. Eric, are you willing to put up contact information if people want to follow up with you and get a little bit more information about some of the procedures that you used?
Absolutely, yeah, sure.
Do you have that on one of your slides, or maybe you can put it in the chat for folks?
I sure can. I'll put it in the chat. Let me figure out how my keyboard works.
Sorry to put you on the spot.
No problem, no problem.
I was just thinking that Karen, who introduced you, was talking about using some of the things in GitHub. And so just encouraging folks to explore what you have produced, I think is wonderful.
Yeah. And
right.
we're very hopeful this paper will come out and in next year, 2021. So there'll be more details on that in the near future.
Nice. It's amazing to hear you talk about next year and then realize that that's just a few weeks away.
I know.
All right. So you it'd be nice for a change. Hopefully we'll see something different in 2021. So you can see, but I'll just let everyone know that Eric is getting lots of congratulations and great talks coming in on the chat. And so if you could hear us all, Eric, we're giving you a great round of applause. And thank you so much for joining us. I really appreciate it. Lisa, did you want to add anything? You're welcome to unmute yourself.
I'm looking forward to hosting you in person up here, Eric.
Can't wait, Lisa.
Yeah,
Thanks
we're
so
excited to keep the collaboration going for sure. Okay, for everybody online, thank you so much. I hope you join us next week, same time, same place, to learn a little bit more about animal migration. If you are curious or would like to contact Eric, he did put his contact information in the chat, so you can contact him directly for any follow-up. And like I said, give me until about Monday, and I should have this presentation up on our past seminars page. So if you missed something or want to look at something again, it should be there and you can review it. And for Eric, thank you again so much for joining us. We really appreciate you taking the time to spend with us. And tell your significant other it was nice to be serenaded as we were welcomed onto our seminar today.
Well, thank you. I really appreciate the time and hosting, and I can send a copy of these slides if you want to distribute those as well. I'm happy to do that. Yeah,
if you want to send them my way, and then folks can either reach out to you directly or reach out to me if they want a copy of the slides.
Sounds good. Really appreciate it. Thank you very much.
All right. Thanks, everyone. And thank you, Eric. Good night, everybody. Have a good evening. I'm going to end the presentation. Thanks, everyone.